Anyway, so how's your research going?
Oh, pretty good. Yeah, I'm kind of keeping up with things here and there, and actually, we're doing some papers at another like a couple of conference submissions that we're working on the last couple of weeks. So it's been pretty busy. This is a different group that I'm working in, so.
Oh, okay. Yeah, well, I'm supposed to be sticking to the one thing.
Yeah.
Well, I tried to get my daughter to make the other gizmo that I was working on, so it's a 3D printed geodesic dome thing, just sort of, not very big, but it's to hold the microscopes.
Oh, yeah.
So I'm making this geodesic dome type microscope, and I handed my proposal in to Dick, and he suggested that I do a comparison with all the other ones that he'd done and the other fellow in Waterloo had done, so we can make it into a kind of complete paper about how we tried to make 3D microscopes, all the different ways. So yeah, there's advantages to each of them.
Yeah, definitely.
Anyways, I hope this will eventually get printed. I'm going to print one ring. I'm going to start by printing one hexagon of the microscope, and haven't got my living room rearranged so I can operate my 3D printer yet. So that's what I've been doing.
Yeah.
In my spare time.
Yeah.
Yeah, I'm learning mechanics of solids right at the moment.
Okay.
Various ways. edX has some nice mechanics of solids, but I'm actually taking a course at the University of Manitoba at the moment in it, so
yeah,
learn the basics first, and then I can continue.
Yeah, it sounds great.
Okay, has anybody else decided to join us? Or?
I got an email from Krishna. He said he may not be able to make it. He's got network issues. I know Dick will be here because this is his alternate week, and I don't know if anyone else is going to show up. So,
okay.
But we could start, and then we could start, and people could join us as they come in. I mean, they probably will start to come in maybe a little bit late. Sometimes people show up. So welcome to the meeting. If you're watching this on YouTube, let me know afterwards if there's anything you find interesting. Today we're going to have Susan's going to give a talk on her work, and I don't know if Krishna is going to be able to make it. He was going to give a talk as well. We'll also talk about maybe a little bit about the EndNote bibliography, about bibliographies. We'll talk about the task board, and then a little bit about the JOS paper, which I kind of put aside for a while because I've been working on some other things, but I'm going to try to work on it a little bit more and get it out soon. So, Susan, why don't you start, and we'll see who shows up at,
Okay, sure. So present now, is that how you do it on here?
Yeah, present now.
Okay, present. My entire screen? I guess.
Yeah.
Why not? Okay, share. All right, this hasn't changed. So how do I do this now?
Well, it should be if you go into the present now, and it should be the entire screen, and it should let you. You could try sharing a tab. So the other option is a window. You could share the window in which it's in. That might work as well.
Okay, well, I'll cancel this then, because it doesn't seem to want to work. So we'll go present now and a window.
Yeah.
Right, okay. Share. Lovely. My computer is not the greatest. All right, so I'm trying to share it, and it's not. Do I do this? Share. Okay.
There we go.
Oh, all right. Just have to press the right button.
Yeah.
So, so I should make this larger. I'm going to stop sharing and try the whole screen again. Sorry.
Yeah, why don't you try the whole screen?
Entire screen. Okay, and then you click on that and you say share.
Yes. Here we go. It's coming up. Good.
Oh, it's still half written.
Well, it's just showing your screen, so it should.
Yeah, okay. So
Can you pull
I
up your
need
slides?
to find this. Okay, can you see that?
Yeah, it's good.
Okay. So I will present this. This is kind of neat. It looks in space. At least I kind of liked it.
Yeah.
Anyway,
I like the star field. It's coming out.
Yeah, this is an older paper of mine. It was written in 2006, and it has since been cited over 100 times,
100
more
times?
recently. So I guess it's striking a chord with people in this area. So I decided to present this since well, you've seen part of my thesis and it's not really organized yet. So this was I presented it at IEEE Women in Engineering lectures out of the University of Manitoba. So that's why it's got University of Manitoba written on it.
Great.
And so when you're in microgravity, you have problems with convection and surface tension and buoyancy and sedimentation. And this is a candle in Earth gravity and work properly. And this is a candle in microgravity. Can you see my mouse pointer?
Yeah.
Okay, and it goes out. Unless you blow on this, it goes out. And here's some more. This is thermocapillary action in, this is Earth gravity, microgravity, and then this is Earth gravity, and this is microgravity. And you notice that the water vapor collects around the heating element. So things just don't work the same in microgravity. Chemistry and chemistry of cells. So this is the cytoskeleton of cells. There's microtubules in, I think it was green, intermediate filaments in black. Oh, okay, microtubule is this blue. Anyway, the blue in this picture, that's the, and then the microtubules are in green, and the microfilaments are the red around here. And this in microgravity. So here you've got Earth gravity. This sample and this sample, these are cuvettes with microtubules being nucleated. And this is simulated microgravity. You notice the pattern is not nearly as rich as it is in 1G, even though it's a simulated 1G. And then you, if you're in microgravity and you simulate 1G, you get the pattern, and it's similar to this. I still don't think it's as rich as what you have over at the beginning. But if you leave them just in microgravity, you get no growth. So your cells have to work at it to create their usual environment. So this is ground control, or one gravity here, and this is microgravity, and the microtubules are clumped and rearranged, and these are lymphocytes, so it's part of your immune system. And that is a problem with astronauts. They do recover within 24 hours and could operate normally, but I think it takes more effort to do that, more energy. Collagen gel. This is Earth normal in this line on the left, and this is in microgravity on the right. And there's a huge difference in texture, and this makes a difference in the strength of collagen gels. So Bradley, are you still here?
Yeah, I'm still here.
Okay, I'm going to say goodbye to my daughter. She's leaving for Regina.
No, actually,
Okay.
I'm leaving for Regina. Dad and I are going to go for a little airplane ride.
Oh, okay.
So
They're
we'll be back
off.
at around 10:30, I'm guessing.
Okay, she's going off with. Okay,
Okay.
good. All right. Okay, that figured out what's happening with my
Okay.
family. Thank you. I know this is being recorded, but anyway.
It's okay.
This is the main image that I had. I can describe the whole paper basically from this image. So you have cells on Earth down here. This they are supposed to look like. And this is cells simulated in 1G in space. They look different. It might be due to the fact that you go on a trip to get there. I don't know, but they look different. And then this is in microgravity in space, and they definitely, all three samples look different. And when you have cells that are not in place, they can undergo apoptosis and die, or turn into cancer cells or into another type of cell. So this is a concern. And then there's your microtubules. I've described that. And this is the difference in gene expression. So in microgravity, you can see there's quite a difference in gene expression here. And in simulated microgravity on Earth, there's some spread, but it isn't the same as actual true microgravity. And maybe part of this is slinging it around in a clinostat, so there's more work on. So that's the summary of it. So if you're trying to reproduce in space, you've got microgravity and space radiation, and they've tried this, I believe, with rats, and it didn't work. So they've tried it with salamanders, and it sort of worked. So you have the salamanders, and I believe this Earth normal. Yeah, these are Earth normal in the right side, and the left side, this is what happens to microgravity. And you get some of the yolk granules from the bottom drifting out, which is not great. And you don't get cell division occurring where it's supposed to occur. And you end up with at neural tubes stage, they said some of the cells were drifting off, like some of the brain cells drifted off into space. The whole thing didn't work that great.
Well, do they raise them in like a? Do they raise them in like a chamber, or do they have?
Okay, I should go back to that paper and try to figure out what they were doing. But salamander eggs, you can just put them in water and make sure they've got oxygen and that they're not too hot, and they will just grow.
Yeah.
So they're fairly easy to handle compared to a lot of other things. So that's they were an experimental animal. But I looked at these, and I believe this one here, some of them, there were multiple problems with neural closure with these. And the one that I think it was this one. I took one look at it, and I'd been dealing with salamanders for a while, and I said, that looks cross-eyed. And it's got microencephaly, so small brain.
Right.
Yeah. Here's somebody's experiment with, this is microgravity created on Earth, so it's they just rotated it so that it was in continuous free fall. And then this is your normal development here. And you can see that it starts off not the same, it doesn't look the same, and it proceeds to not be the same, and the final developmental stages. This is microgravity, this is normal, and this is 3D. And so all of that caused me to say, well, if I was and you were the spaceship captain, be going to your office and approaching you and saying, I want 1G Earth normal gravity now, or I will sue you. Just from looking at this.
Yeah.
Yeah. So it's a problem. And this is a more recent paper. And they were looking at oocyte, that's the mice eggs, as they were developing their polar bodies. This is Earth normal on top, and this is microgravity. And so you can see there is a difference.
Yeah.
The microtubules in green, and the nuclear matter is in blue. And this develops normally, and this looks like it's dying to me.
Yeah.
So this is at 6, 8, and 16 hours. So yeah, it doesn't look promising. Not even from the get-go.
Yeah.
Not even to produce an egg. Never mind anything else. Anyway, that's my talk. I have my papers online for free. And then this newer paper, it was also online for free, I believe.
Okay. So the 2006 paper is your paper that you were talking about at the beginning.
Yes.
Okay.
Yeah. Mm-hmm.
All right. That's good. I'm the only one here to ask questions, I guess. So,
Yeah.
I guess I had some questions about like how, so they raise these embryos in space, or they raise them at like Earth gravity, and then they raise them in space, which is reduced gravity. And so in space, you see all these sort of abnormalities of morphogenesis where they have different, shape deficits or basically the shape gets warped somehow. So gravity must be playing some sort of role in mediating the shape, or is it like maybe something like that they're receiving cues from? Gravity changes, but do they keep the water pressure constant, the air pressure constant, that sort of thing?
Yes, that's normal. It's just the thing they're changing is gravity.
Okay.
And it actually changes. I don't know if you know what EZ reaction is.
Yeah.
But, that changes. Like the timing of a big reaction will change in microgravity. It affects diffusion. It affects, like I said, sedimentation. So and if it's like that convection, like microtubules are partially could be formed from convection, and there's no convection. And so it changes the way things are done. Right. Chemically, physically, and otherwise. So And there's never been mammalian development in space. There's no reproduction of mammals in space, as far as I know.
Right.
They have negative results, and they don't like publishing.
But the salamanders do. They can.
Well, yeah, they end up with thin brains, sized brains, microencephaly. They end up with abnormality.
Is that because they have they have a regenerative capacity, so they can overcome a lot of the things that might kill a mammalian embryo? Or
No, I think they're bigger, and they also don't require as much they're not as reliant on microtubules and the cell cytoskeleton to get them going. Go back to what I presented earlier, where there's extreme pressure inside the developing embryo at first. Like it was like fracking. There was so much pressure there. And embryos have to hatch. They have to hatch to their surrounding eggs. It's called the spinopellucida. But we have to hatch out of that in order to develop. Like you can't just take our little embryo out of that and have us develop. We don't. We need the pressure in order to rearrange the material inside the egg so that we can develop. That's how I look at it. And that doesn't occur properly in microgravity, especially when you've got a 10 to 70 micrometer egg that you're dealing with. It's very small. Salamanders are big. They're very hardy compared to us in a number of ways. And that is one of them.
Yeah.
They just have dysmorphologies. So whether how well they would survive in the wild, I don't know, but they at least they hatch.
Yeah. Well, that's pretty good. Thank you for that talk. Can you share your slides with me? I can share them with the rest of the group.
Yeah, I'll make a PDF, or I've got a PDF of that.
Okay.
Okay, so now I just need to stop sharing. There we go.
Yeah, there we go. Okay.
Oh, good, just in time for.
Yeah.
Sorry about this. I'm going to try to turn off my.
Yeah, just mute.
I'll turn this off. I don't know if this is going to work, but we can try.
Oh. Okay. So thank you, Susan, for your talk. And I've got a couple things I'd like to cover. Oh, there's Rish. Oops. I think I pressed the wrong button. I was going to let him in, and I don't know what happened. So he'll come back, maybe. Well, anyways, let me go to what I was going to talk about here. So I had a couple things I wanted to talk about here. And if Rish comes back, we'll have him introduce himself. And so the first thing I wanted to talk about was this bibliography, these bibliography materials. So this is something that Dick and I have been working on in EndNote. So EndNote is, of course, a bibliography method that we have for organizing references. And so one of the things you'll learn if you do research, and some of you do research and some of you are starting out, is that you need a good method for collecting references and organizing them. And so we've been working on this EndNote bibliography for this. This happens to be just an example from a project we're working on where we're looking at something called the Boring Billion, which is a period of evolutionary history before the Cambrian explosion. And so we have all these references for different papers in that topical area. And we're using EndNote to organize them. Now, when you organize references, you can often go to like a database, say like Web of Science or some other database, and do a search, and then import the references directly from that tool. So these were downloaded from the Web of Science. These were based on a number of keywords. So when you do a literature search, you want to have a list of keywords that you're working on, and you want to kind of make a record of what those search terms are, and then you want to get your results that you get from that search and save it. So I'm not going to go through the steps required to save something from, say, Web of Science or some other source, but I will say that you can do that. You can have a search completed and then download it in a bibliographic format, which is usually a .bib file or something similar. And then you can take that file and import it into your one of these, and EndNote isn't the only solution, but one of these bibliography programs. So EndNote is a proprietary platform. It allows you to collect a large number of references and organize them. So here we can see that we have a alphabetical organization of the references here. So if I go into this, all my references, I get 45 references and they're alphabetized. But we can also do things like put tags on them. We can organize them by groups. So I have two groups in this bibliography. I have Boring Billion and I have cell tracking. And so cell tracking is a little bit smaller group of papers, and it's different from the Boring Billion stuff because it's not the same topical area at all. So, in this bibliography, I've just saved two different groups, one being Boring Billion and one being cell tracking. And, the benefit of this is that, if I have something that's not really related to one project, I can switch over and get a label for the other project. Here's Jesse. Hello, Jesse.
Morning. Sorry it's late.
Yeah, it's okay. I think I saw Rashab coming by, and I hit the wrong button to let him in. So maybe you can let him know that we're to try again, because I don't know why. Yeah, so you might rise him on Slack or something.
Okay.
All right. Otherwise, yeah, welcome to the meeting. Susan just gave a talk on low gravity cell culture, which was really good. I'll share the slides later. So
Yeah, I'll look at that.
Okay.
I'll check that out. Yes.
All right, yeah. So I can go back to the EndNote thing. So this is basically the way out of EndNote, and so we've talked about doing this in EndNote, and in my other group we've talked about doing this in Zotero, and so that's basically, you have a number of options for doing organizing bibliographies. Zotero is a freeware that you can use and you can plug into your browser, and you can import references from the web. So you can actually take references as you encounter them on the web and put them into the bibliography. I think you can do that with EndNote as well. So you don't have to go to a formal search engine like Web of Science isn't really a search engine, but a formal search tool like Web of Science. You can actually take them off the web, and most people encounter references sort of on the web as they're searching. That's another mode. I actually also use Mendeley, which I'm not going to show you right now, but Mendeley allows you to take PDFs, put them into a window, and then the program extracts all of the metadata, and so you end up with a citation like this with the PDF file attached, and it goes right into the bibliography that you have. You don't have to save files. It's sometimes these programs like to save PDF files, but there are a lot of problems with that with respect to like storage and sharing these things. So we like to assemble them a lot of times without the PDF file attached. And so why do we want to do this bibliography thing in the first place? Well, I think it's a good idea to have first of all of our um, the things that are published from the group in a bibliography, but also all the things we've encountered. So all the papers we've published, we can get a bibliography from each paper and put it into a reference manager like this, and then we can share it with new members of the group, and they can see kind of what the literature looks like. And so that's one reason you might want to do it. Another reason you want to do it is because you have a record sort of what you've already done, so you don't have to duplicate work. If we want to follow up on a topic, we already have a list of references that are pretty well known to the group. In my other group, we had a discussion, I think Saturday, about taking these like you go through a paper and you have maybe 50 references, and you might have to go revisit a couple of those references to, like if you're reading, we're revisiting a paper right now in the other group where we're taking like some angle of that paper and we want to write a new paper on it. So we have to go back to the reference list and we have to find those references that were most important for that certain part of the paper and then follow up on that. So there are a number of reasons why you'd want to use a reference manager. So this is where we're working with EndNote. We're working also with Zotero. We're working with our references for different papers, and hopefully we'll have more to report on this in future meetings. But I just wanted to give people a taste of that because I think it's something we don't talk about very much, but I think it's an important thing for managing research. So next thing I want to turn to is this, and we haven't done this in a while, the project board for DevoWorm, and this is for the group meetings. So this is the link to the project board on GitHub. And again, if you remember last time we did this, which was several weeks ago, we just kind of went through and looked at what we have in terms of major tasks. And this is actually pretty incomplete because we have a lot of papers that we're working on, and I don't have these in here. So it's going to be a little bit tricky to, this is like these are odds and ends aside from the papers that we might want to follow up on. Here's Ujjwal. Okay, hi Ujjwal. So the major tasks, I went through this last night and I looked at some of the things that we have. So we've got a lot of things finished on this board. Presented a number of ideas. We've gone through GSOC. We've done some stuff on agent-based modeling and this Neuromatch submissions, which we did submit a abstract for Neuromatch. I'm not going to talk about it today, but the session is coming up in a couple weeks. So I might send out some materials on that in the meantime. The Neuromatch submission has to do with the Basilaria model, the psychophysics model. So I haven't talked about that too much in the group, but basically it's this idea that the Basilaria colonies exhibit some sort of psychophysics. And it's going to be a very simple model that's going to be presented in a 15-minute presentation. So I don't even know if we're going to have a meeting between now and then because next week I have an event I have to do. And then the week after is Neuromatch. So I think that's going to be, I'm not going to be able to show that before the meeting, but we'll go over that. We'll talk about that more in the future. And then maybe we'll talk about it offline as well. So we've got this lecture on PCA, UMAP, and TSNE. So if you missed this last week, this was a lecture I gave on basically multivariate statistics and how to interpret them. It was a lot of visualization and how to, it looks like a pretty picture, a lot of these multivariate methods, but what do they actually mean? And that was an interesting lecture. This revisions for bacillary morphology paper, that's old, but we did that. That's accepted. Some more ideas that were presented on. And then we talked about the CC3D thing, which we put aside. Okay, so we have in progress, things that are in progress. So Hacktoberfest is going on right now. And so we talked about Hacktoberfest last week. We had a flurry of contributions early, which seemed not to, we haven't had any contributions this week, although we had an inquiry in Slack on something in the DevoWorm channel. And I don't know who wants to handle that question, but it's in there. So, but other than that, we have a number of people who have already contributed to Hacktoberfest. And so I put those people, I sort of acknowledge them in the weekly email. I'm going to try to make a blog post for Hacktoberfest soon, where I kind of go over the idea for Hacktoberfest and how to contribute, and then to acknowledge the people who have already contributed. So that'll be more public facing soon. But here's the bibliography issue number 48 for EndNote. This bibliography issue is still open. So I was showing you the work that we've done so far, but we're trying to work on, finalizing some sort of release for this in EndNote. And then this will be, you can convert this to some other format if you prefer, for example, using Zotero or some other bibliography thing. We can make versions of that as well. But we're going to assemble the bibliography in EndNote. The Lagrangian embryo, that was just like kind of a presentation, and that didn't go much further. But if you're interested in this idea, we need to assemble some readings. So this is something that we might do if people are interested. Recruit people as DevoWorm contributors. I think we did some of this for Hacktoberfest. We had some people come by. I think the blog post will recruit people into this a little bit. But, I think there will be some interest as time goes on with DevoWorm, people encountering it. And I presented on DevoWorm at the annual meeting, so that's there's some interest there. So we'll be following up on this. This is, I don't know if this is a duplicate here, bibliography and EndNote, but 59 and 48 are linked. It's the same issue, more or less. Create a theory layer for DevoWorm. This is something else that we've talked about doing, so this is kind of, maybe we'll put this in hold, where we just create like a sort of a theory building mechanism within DevoWorm, like we have for the data science part. I know there was Jesse, I think, contributed in the education area, so that's something else that we're working on, and so we'll follow up on that as well in future meetings. This periodicity in the embryo papers is something that I think we need to pick up on a little bit more. At the end of the year, there's so we're trying to submit to the special issue of Biosystems, and this paper, I think we have a draft due at the end of the year. So this draft, I guess it should be just kind of like a rough draft. So it's more than an outline. We're doing maybe, we should maybe aim for like 10 pages or something, maybe 12, where we can get a good rough draft on it. And we've talked about this paper. This is the one where we have the different model organisms, and we're looking at the different periodicity components of embryogenesis, things like cell division, cell differentiation, things like that, and maybe creating graphs or different types of analysis for it. So, I put this aside myself, and I know it's not been like something that we've really has been on the front burner, but this is something we need to revisit. I think in the from now until the end of the year, because we're going to have a rough draft due at the end of the year on this. Then linked to that is complexity measures, which is this idea of different sort of measures of complexity. They could be mathematical models. They could be different ways we can characterize complexity. And this is, of course, linked to this periodicity and the embryo stuff, but also the boring billion, which I don't know if I even have on the task board. But this is something that it's kind of one of these very large issues that, get chopped into different pieces over time. So, and then finally, the bacillary non-neuronal cognition paper. So this 4 is actually linked to this Neuromatch abstract here that's already been finished, but we'll use that to sort of continue on in the direction of this paper. And this is another paper that's due maybe early next year. So we have a bit more time on this one. But it's still, yeah.
I was going to ask about that one. Is that if that was related to the Neuromatch submission or not? Because I was wondering. Also, I mean, I guess also, I wanted to say I saw the kitty on the camera, and that was a good thing earlier. But yeah, between that and the complexity measures paper, which I think I was looking at a while ago, the non-neuronal cognition paper was something that I was hoping to. Is that one of the open? Is that the same thing as one of the open papers that's listed in the open papers in the GitHub?
Yeah, it is one of the open papers. So, I mean, my thinking about that submission is, where we do some work for the Neuromatch submission, and then we have that in place. There's the kitten, and yeah, and we then take that and use that as sort of like a kernel for the analysis for that paper. So, I mean, it's going to be, we want to build in steps here. We don't want to just, do a brain dump at the, near the deadline. And then we want to kind of this is why we kind of want to do conference submissions if we can, or other types of things where we can put the pieces in place. And so we haven't been able to do that with the periodicity paper, but I think definitely that's a good way to, it's always a good way to have a stepwise sort of progression like that. You come up with some piece of the paper, and then, you can write around the main idea more easily.
Okay. Yeah. That's pretty much what I thought. I wanted to make sure. Thanks.
Yeah. So, okay. So Ujjwal says, next week I have my mid-semester recess, and I've already planned to work on creating 3D models. I will surely show some results on 26th October if we have one. Yeah, 26th October meeting, we might have one. This is the week of Neuromatch, so I'm not really sure. We don't have our schedule for the talks yet on that. But if we don't have talks scheduled, I probably will have a meeting that week, and we can do it then. Because I'm already going to miss next week because I have another engagement during that time. So we'll talk about that more. But yeah, we can plan for that, I guess. So there was an issue for that. That was, I think, 38, where we have the create an embryo model in Blender. And so this is something that Ujjwal's been working on. He started in CC3D, and then it wasn't feasible to do it that way, so he was doing it in Blender, which looked, pretty good. He had some good results the last time we checked in. Yeah, no problem. We can get some results in Slack and then do it in the meeting and, kind of move forward on that. So this is also something that we can potentially contribute to this Docker container. So again, the Docker container, we revisited that during the annual meeting, and they're still interested in something from DevoWorm, but it's something where they have like a little simulation or a little visualization from every group in Devo or in OpenWorm, and so that would be good. Let's see, yeah, and then there's some issues for Krishna. I think he's still working on them. The general biological model. Krishna actually submitted something to Neuromatch on the difference between biological neural networks and artificial neural networks, which is something that we'll be working on sort of offline, and then maybe we'll present on in this group later. Let's see.
Yeah, I'd like to see that. That was interesting. Neuromatch submission, and I'd definitely see more of that here too.
Yeah, we'll probably talk about it in the meetings. Then there's the axolotl montaging and axolotl segmentation stuff. So that's issue 5 and 25. I put that under Hacktoberfest because it might be something people are interested in for doing, Hacktoberfest, incorporating that into the DevoLearn repositories. We're still kind of, I guess, that's still kind of open. And so the kitty again. And then so that's good. So the axolotl stuff, we're still kind of open on that. And then to do, we have things. So Jesse mentioned this paper review of scale-free biology. That's something maybe we'll do in the future. Let's see, anything else? Not really. I mean, these are just issues that are kind of, like I said, I put them up as odds and ends, maybe somewhat independent of the paper plan that we have, but they're things that we should check in on from time to time. So I also wanted to mention this Joss article. So this is the one that I've been working on with Mayuk, and this is where it stands right now. So we have a summary, a statement of need, and then references. I'm going to work on this a little bit more and add some things in. I see that Mayuk last night added in a PDF of like a description of the project. So it's largely what he put in the notebook, in the Jupyter notebook or the Colab notebook. I can't remember which format he used for his project. And so we might pull from that. And there are some other motivational things I'd like to bring up in here. So I'm finally turning back to this after working on the some other submissions for the other group I'm working in. And Jesse knows very well what I'm referring to. Yeah. Okay, so that's the Joss paper. That's on the hot, sort of on the front burner. Finally, I'd like
that paper
open?
Like, is that? Are you looking for other people to work on that, or is that kind of a set piece for now?
I think it's probably a set piece. We've got it's going to be very simple. Like it's a simple description of the DevoLearn software package. So it's just kind of like,
Okay.
But I just need to add a little bit more to it. It's the Journal of Open Source Software. If you don't know, is this open? It's a completely open source journal that is basically run. You take like your piece of software that you've created that's open source. You wrote a little bit of maybe about 1 or 2 page description of it with some broader implications, and then you put it on GitHub and you send it to these people. Yeah. Thank you, Ujjwal. And then you get, they review it and they say, this is good. I mean, it's basically as stripped down as you can get for a journal, which is it's really fascinating. You have like this, you just have editors and they like go through a paper and they see if it's good. And they're very stripped down descriptions of the work. And So I mean, it's an interesting journal. Hopefully, we can get this in because this would be a nice summary of the Summer of Code stuff. So maybe I can get Ujjwal involved as well. But I'll send him a link to it. Like I said, it's largely about the DevoWorm thing, but we might actually mention a little bit about the platform that we've been developing. So, we call the program DevoWorm, and then we have the platform that's DevoWorm. And it may be a little confusing, but it's, one has a version number and the other one is a broader thing. So, we might talk about the broader thing a little bit too, just to show like how this fits into a larger scheme. So, any questions about anything so far? I wanted to talk about one more thing before we go, but Susan, you're muted.
No, there. I'm very interested in the axolotl 3D montaging, and it would go with the paper that I'm writing currently on the other 3D imaging microscope and the review of the other 3D microscopes. So if someone were to do something on that, it could be part of the paper.
Yeah.
Or a complementary paper, whatever. Yeah.
So you're actively working on like another paper for the, you did the paper on the flipping microscope.
Yes.
So
and then I'm letting my daughter take over for this particular project, and she's lead author because she's doing the 3D printing and setting it up and everything is just sort of basically Dick and my idea of getting a 3D microscope, and then if someone could do the montage, then it could be a part of what we're doing.
Yeah.
So I just wanted to mention that.
Yeah.
Like it's actually a paper in the works that could incorporate that if anybody's interested.
Yeah. Well, yeah, I'm trying to get like, I mean, it's slow moving, but it's like we still want to make sure because, we'd like to make it into something that we can also incorporate into the DevoWorm platform, or at least people can use it, and so.
Well, I'm hoping that other types of cells besides an amphibian egg could be imaged using this idea. So.
At the general meeting, I did put a slide in on the axolotl stuff, and someone talked about Xenopus as a potential thing that you could look at with that microscope setup and everything.
Yes.
Yeah. So I mean that's that's something that's probably in demand. I mean, people would like to see that, I guess. So.
Yeah, there's other types of amphibian eggs as well. Like if you do a comparison of how they develop. I have some old 1950s papers on that, which are interesting. So, yeah, studies been going on for a long time, and I think this would, it would further developmental studies. It doesn't matter whether it's Xenopus or any other type of, salamander or frog or toad. Anyway, people have studied them for a long time.
Yeah.
Okay. That's all I wanted to say.
Oh, yeah. Good. Thank you. So I think that's a good thing. We'll have to follow up on that. I keep bringing it up, but we're kind of moving a lot more slowly than I'd like, but that's okay. I mean, that's always the way it goes. You just have like a lot of things that come up, and but I like to go revisit things, make sure that we remember them. They don't get swept under the rug and forgotten about, because that often happens in research. So, the last thing I'd like to talk about today is I have some papers, and I haven't talked about these papers for a while. I haven't had session where I've talked about papers. I have 4 papers, but I'm just going to kind of talk just maybe a little bit about them, just to give you, again, a little taste of newer papers that are coming out. So this is an older paper, but this is something that I ran across that's interesting. So this is called Dynamical Patterning Modules, Physiogenetic Determinants of Morphological Development and Evolution. And so this is a paper about the shape and form of multicellular organisms and their development. So the argument here is that your shapes and forms that you see arise is sort of the product of new cell states that come about and changes in the numbers and rearrangements of the various kinds of cells. So that's the way they think about it. They're thinking about it in terms of different types of cells, the origin of different types of cells and their rearrangement. So while morphogenesis and pattern formation on all animal species are widely recognized to be mediated by the gene products of an evolutionary conserved developmental genetic toolkit, the link between these molecular players and the physics underlying these processes has been generally ignored. So we talked about the physics of embryogenesis earlier in the meeting, and we've also talked in prior meetings about the genetic underpinnings of, at least of cells differentiating. And so then their idea here is to sort of bring them together. So this paper introduces the concept of dynamical patterning modules, DPMs. And that is a unit consisting of one or more products of the toolkit genes that mobilize physical processes. So this is characteristic of chemically and mechanically excitable systems. And these include cell aggregates. And you see these cell aggregates in action in terms of cohesion, viscoelasticity, diffusion, spatiotemporal heterogeneity, multi-stable and oscillatory dynamics. So basically, a lot of the things you see in cell collectives, their physical properties, but also these sort of they're almost like neural systems. So like the lateral inhibition, there's an issue here with spatiotemporal heterogeneity. So if you have different cells within close proximity and they're in this mass, they have an influence on one another. And so in this case, they're talking about maybe lateral inhibition being one of those influences. Furthermore, you have multi-stable and oscillatory dynamics. So like in brains that fire action potentials, different cells have different types of dynamics internally, and that has some effect. And so they suggest that ancient toolkit gene products, most predating the emergence of multicellularity, assume novel morphogenetic functions due to changes in the scale and context inherent to multicellularity. So this, again, is that multicellularity had this sort of transformative effect on the cells and the way they behave in these aggregates. I mean, you had, before multicellularity, you would have cell aggregates, but there's something about multicellularity that was fundamentally different. And there's a lot of literature on this. They call it a major transition in evolution, where you start to get these multicellular or multicellularity as a thing that you see in nature. And it's much different than just a cell aggregate, because there are a lot of community effects that arise around true multicellularity. So they show that DPMs, acting individually and in concert with each other, constitute a pattern language capable of generating all metazone body plans and organ forms. This idea of pattern language actually comes from architecture. It was Scott Alexander, I think, who proposed a pattern language for, and it was really popular about maybe 30 years ago, where they talked about it in programming and they talked about it in architecture and in design. And it's like, basically combining patterns in a certain way that's, has some sort of linguistic value or some sort of semantic value. So I don't, you have to read the paper to find out more about that, but it's an interesting take on this. So another point that they make here is the physical dimension of developmental causation implies that multicellular forms during the Cambrian explosion, which was one of the first events in multicellular life that really resulted in this proliferation of life, approximately 530 million years ago, could have explored an extensive morphospace without associated genotypic change or selection for adaptation. So if you know anything about the Cambrian explosion, you know that you ended up with only a couple of body plans that really proliferated. Despite all of the different forms that came out of that period, they come from a handful of body plans. And so those body plans, though, may not be they may not really be that restrictive. They might be representative of maybe the best of a wide range of possibilities. And so we don't know that, but so yeah, if you want to find out more about this paper, I'll give you a link to the drive here. And so if you want to know more about that, you can read the paper. The second one is this before programs. This is another Stuart Newman paper. This is from 2006. This is the physical origination of multicellular forms. And so this kind of is a follow-up to the last paper I told you about. And I think it's work that sort of leads into the other paper that I just presented. So they do some work on, by examining this formative role of physical processes in modern-day developmental systems, we infer that although such determinants are subject to constraints and rarely act in a pure fashion, they are identical to processes generic to all viscoelastic, chemically excitable media, non-living as well as living. So basically they're saying that they're like what you see in development in terms of morphogenesis is very similar to what you see in any sort of chemically excitable system or viscoelastic system. So this, then there's a distinction between whether it's non-living and living. Actually, I found a paper recently. I don't know if it was submitted to one of the machine learning conferences, but it's where they're actually simulating like flows and other sort of physical processes like this. And they're trying to determine whether it's a biological process or a sort of a inorganic process. It's kind of an interesting paper. I don't have the link to it right here in the meeting, but I think it's kind of interesting that these things are related. So
I'd be interested in that paper.
Yeah.
Just the one describing. It's actually very related to the research I'm doing.
Yeah, I'll have to pull it up. So that's the newer paper. This one, though, they talk about sort of how morphogenesis that we observe in embryos came to be, and so they kind of go through this. So from this, we devise a hypothetical scenario for pattern formation and morphogenesis in the earliest metazoa. We show that the expected morphologies that would arise during this relatively unconstrained physical stage of evolution correspond to the hollow, multi-layered, and segmented morphotypes seen in the gastrulation stage of embryos in modern-day metazoa. So this is like they're trying to get an idea of how this, if you take this sort of inorganic process and map it to this organic process, what are the evolutionary origins of it? And so it's an interesting paper. Again, it's in this folder. And maybe I'll put that paper that I was talking about in this folder as well. The third one is quantitative approaches in developmental biology. So this is a review it's another modeling review. This is, again, like several weeks ago, we talked about the agent-based modeling approach. And so this is kind of a different take on it. This is modeling development, modeling developmental biology, but taking a more general quantitative approach to it. So the tissues of a developing embryo are simultaneously patterned, moved, and differentiated according to an exchange of information between their constituent cells. We argue that these complex self-organizing phenomena can only be fully understood with quantitative mathematical frameworks that allow specific hypotheses to be formulated and tested. The quantitative and dynamic imaging of growing embryos is the key experimental advance required to achieve this interaction. Here we describe how mathematical modeling has become an invaluable method to integrate quantitative biological information across temporal and spatial scales. So they're really kind of focused on imaging data and how to pull it together using quantitative methods. And I'm not really able to manipulate this file, so I can't go down any farther into the paper. But it's basically, the approach is very similar to things we do in this group. But they're kind of reviewing. Okay, we can open it with something here. I just want to open it up and look at it. Yeah, let's do Google Docs, I guess. If it opens. I don't think we're going to do that. Let's try this one. We'll try PDF filler. Oh, no, forget it. It's okay. I don't want to go through all that. That's okay. We'll just, if you want to look at the paper, it's in this folder. And I think it'll be really interesting because they kind of go over, it's quite a bit different take than the agent-based modeling, in the sense that it's really kind of based right on what you're observing from the data. Now, that being said, a lot of these quantitative models have shortcomings that agent-based models can fill in the gaps. And one of the big ones there is the generative aspect of development. So we can observe things with, microscopy data, say, and we can take those data, extract them, and analyze them in a quantitative fashion. So we can extract things out of the images and put numbers on them. But that only gets us so far. It only gets us to what we can observe. But we can't observe every aspect of the pattern. We can't observe every aspect of what's going on or what could happen. And so that's where your agent-based models come in. It gives you that perspective of, the generative aspect of development, where you can ask questions that are maybe more rooted in hypotheticals than what you would get with just observing data. But nevertheless, we do need to have good quantitative approaches. And so that brings us to the final paper, which is Canalization and Control in Automata Networks. So this is, it's kind of maybe a little bit of a wild ride for some of you, because it's sort of an artificial life type paper. So they explored body segmentation in Drosophila, but they're using this model called automata networks. And they're looking at something called canalization and control. So these are two different concepts that they're introducing here. So canalization, for those of you who don't know what that is, it's something that is common in developmental biology theory. We talk about it sometimes in terms of like phenotypes. So canalization is the idea that development can unfold in a number of different ways, but it tends to go down certain pathways. So if you have an embryo and it's just sort of developing, there are certain paths that an embryo might take in terms of its morphology, in terms of different possibilities that are sort of locked in and conserved over evolution, and then you see this in development. So there's this metaphor of the epigenetic landscape, which is, if you think about like a fitness landscape or a gradient descent landscape, you can kind of visualize what this looks like in your head. And the idea is that it's like this ball rolling down a hill with different, channels and things like that coming down the hill. And the ball will basically go take the path of least resistance and go to places that are lower in elevation. So, it's sort of minimizing its energy. And so the idea of canalization is that these channels represent local minima that guide you through time. And so these channels are where you tend to see a lot of things recur in different embryos. And so they're trying to characterize canalization using these automata networks. Canalization becomes synonymous with redundancy present in the logic of the automata. So basically things that reoccur over time. And so they're able, in this way, they're able to develop measures that quantify canalization, which is turned into a highly scalable framework to characterize the collective dynamics of large-scale automata. So this is a pretty wild ride. Like I said, if you're not used to like this type of work, because it does have a lot of, it's very heavy modeling. There's a heavy modeling component, but they do talk about like different aspects of developmental theory. And so one of them is this idea of buffering. So what you get with phenotypes is you get, so Conrad Waddington is the person who came up with the epigenetic landscape. And so the notion of canalization was proposed to explain why under genetic and environmental perturbations. So when an embryo is developing, they experience all sorts of perturbations. It could be a lack of gravity. It could be, heat, stress. It could be a lot of things. And the idea is that the phenotype can either basically be severely affected by that, or it can be invariant to those stresses. And so when it's invariant to those stresses, you get what they call a buffering of the phenotype or of the developmental process. And this buffering relates to this canalization, in that the buffering keeps the phenotype in these channels. It keeps it going down this minimal path of, this path of least resistance. And so you always go into these channels. That's this buffering effect. And so the buffering effect just basically allows the embryo to survive these sorts of perturbations by finding a common pathway, to development. It's basically, if I, started perturbed, if I put an embryo under heat stress, for example, if an embryo existed in nature where it exhibited severe heat stress, the embryos would all die off and we wouldn't have any, new generation to talk about for the organism. But if they follow this buffering program under times of stress, they can actually survive at much higher rates because, they basically selectively cut out the influence of environment on what's going on with their development. And so that's basically the idea. And so they talk about modeling this canalization, and then they also talk about control in terms of how the phenotype as a whole is controlled in the face of a lot of these stresses. I mean, we think of an embryo as being this thing that, it's almost like mysterious as to how it emerges from this one cell state. And we know that the cells interact, and we know a lot of things about that. But we also know it doesn't have like a macro controller, or at least we don't think it has a macro controller. Although if you read some things on the literature on regeneration, you find that regeneration actually works on a macro scale. In other words, you can have, like in some organisms like flatworms, you can, delete most of the phenotype, and you can still, from the little bit of phenotype that's left, get an entirely new worm that looks exactly like the old worm. And so, in some systems you might have that sort of global control, but it's unclear as to how that all works. So yeah, I think this is an interesting paper, but again, like I said, it might be a hard slog if you're interested in those topics. So that's all I had for today. Again, I'll maybe bring that paper I was talking about and put it in that folder and send it out to the group. So if you want to follow up on this stuff, you're welcome to do so. And also any of the projects that we talked about, again, we can drive any of those forward if we have something that we want to do. If we have any sort of initiative or any sort, if something suddenly becomes relevant, I'm finding that if you have like a deadline, or if you have like something to prepare for, like a conference or something, you can bring some things that you've been procrastinating on or not really paying attention to, and you can bring them to the forefront. So that's always a good way to approach these things. So, but anyways, well, thank you for attending the meeting, and have a good week. Next week we won't be having a meeting. I'll be busy with another engagement. The following week we have Neuromatch, which is again, we're going to have a abstract represented there, but we may still have a meeting just to have a meeting, and then because Neuromatch is going to be going on like throughout the week and at different times, so our presentations might be at weird times, but we'll see about that. Let's see, Jesse.
Oh, sorry. Yeah, this noise here. I was just going to say, I'm very interested in the canalization paper and some other ones today. So hopefully that'll come up again as well. I'll have a phone call coming, so I guess
Okay.
I'll just I'll say thanks to you and see you guys next time.
Okay. See everyone next time.